Dr. Motulsky is a living legend at the University of Washington. He was a refugee from Nazi Germany and was on the notorious SS St. Louis that was bound for Cuba and went back to Europe because no country would accept those on board. Arno found his way to Chicago and joined his father and took an initial job as a student assistant in a hospital laboratory. From there, he did his undergraduate work at Yale, received his medical degree at the University of Illinois in Chicago, and was recruited to the University of Washington as a hematology instructor in 1953. We have to thank Dr. Robert Williams, then chairman of the Department of Medicine, for telling Arno to look at a career in medical genetics. That launched a long, illustrious career, including foundational work in blood diseases, hereditary environment interactions, and the pathogenesis of disease, and the molecular genetics of color vision. But perhaps one of Arno's biggest accomplishments was the establishment of a terrific, incredible training program, making the University of Washington clearly the premier training program in adult genetics in the United States. There is no one who is a more appropriate recipient of the inaugural Lifetime Achievement Award than Dr. Motulsky.
Members and guests, I very much appreciate receiving this award from our organization that represents all facets of clinical genetics. This first Lifetime Achievement Award is a signal honor. Our knowledge of genetics, molecular biology, and other relevant sciences is mounting rapidly, and many applications for diagnosis, treatment, and prevention exist or are on our horizon. The American College of Medical Genetics and its members play a key role in all these endeavors as shown by the program we've seen and that's going on here. Since early in my career, I've been the fortunate privilege to follow my interests in human and medical genetics. As an example, I was fortunate to be exposed to a rigorous course in genetics, including a Drosophila lab as a pre-medical student at Yale University. After medical school, I became interested in hematology, a specialty that deals with many genetic diseases such as sickle cell anemia and other hemolytic anemias that I got interested in studying. As a hematologist at the University of Washington, I gave bootleg lectures to medical students to demonstrate clinical vignettes that were helpful for the diagnosis of genetic disease of all specialties. As was mentioned, the chairman of medicine, Robert Williams, heard all that, and he asked me to start a division of medical genetics, similar to divisions of cardiology and endocrinology and so on. I first hesitated a little because there were no real role models, but there are some, Jim Neal particularly, and I accepted. No, this was in 1957, the same year that Victor McCusick, who I think would have been a great candidate if he wouldn't have died for this particular award. He opened a clinic, a medical genetics clinic, and in 1959, when the University Hospital in Seattle got started, we could start a clinic in medical genetics. Now, over the years, as was mentioned, we attracted at least 70 postdocs and many other postdocs who were involved with me in some way, but worked with other people in Seattle, many from abroad. And we encouraged them with lectures, journal clubs, and conferences, to learn medical and clinical genetics, but also to get involved in a research project with a strong laboratory basis. One fellow, Joseph Goldstein, won the Nobel Prize in 1985 for work in genetic lipid abnormalities that had started in Seattle in this manner. The flowering of biochemical genetics causing inborn errors of metabolism at that time was most exciting. Also in 1957, I wrote a programmatic paper pointing out that enzyme mutations sometimes can cause adverse drug reactions and laid a foundation for the field that later was called pharmacogenetics and then pharmacogenomics. This field was often cited, also now is often cited, in the rubric of personalized medicine. It has become popular, but as currently requires much more clinical investigation. I stress clinical investigation to fully introduce pharmacogenetics into practical medicine. It became clear that better and fuller understanding of the applications of genetics in medicine required work in fields such as statistics, immunology, cytogenetics, and population genetics, in addition to biochemical and clinical genetics, and various other clinical specialties such as neurology and dermatology. Experts in these areas were trained. We helped to train them in Seattle, or were attracted to join our group in Seattle. I then also served on the founder specialty board of medical genetics that helped to formalize the field as a clinical specialty. Ethical and social dilemmas are often caused by genetic problems, as shown also by some of the symposia at this meeting. I was privileged to serve on various policy-making bodies, such as the President's Commission, appointed by President Carter, on ethics in the early 1980s. And in this commission, among other issues, it pointed out the exciting future of genetics, but at the same time also demystified some of the wild ideas about what genetics will do to humans that had been raised by some people. In the mid-1990s, I had the privilege to co-chair an NIH committee which helped to dampen the over-enthusiastic use of human gene therapy before its full scientific basis had been worked out. And it's been taking a long time, and we're just at the beginning. There still is, I think, hope for gene therapy, but it has been slower than we had hoped. Finally, I thank my mentors, colleagues, collaborators, and particularly fellows and students for having made possible the work that led to this award. Last but not least, my wife,